All right, let's get started. This is 5.1 on autoimmunity. This is beginning Unit 5, which kind of covers dysregulation in general. We'll start with this talk, just priming you on autoimmunity, and then we'll go into the different cases throughout the unit. So just broadly speaking, what is autoimmunity? Well, it is a failure of the immune system to distinguish self from non-self. This results in an immune response against the body's own cells. Okay, so obviously that is pretty detrimental and not ideal. This is a breakdown of immunological tolerance. My immune system is no longer tolerating my own cells. So the tolerance is a set of safeguards that normally prevents reaction to any self-antigens. Okay, so I tolerate the cells of my gut. I tolerate the cells of my musculoskeletal system. If for whatever reason the immune system deems those cells to be problematic, well then I stage an immune response against them, and we have a whole litany of conditions associated with that. So every protective mechanism we have discussed can become destructive when turned against the self. We saw that very much so in the case of sepsis, right? There's nothing inherently pathological about vasodilation or increased vascular permeability or neutrophils releasing reactive oxygen species. Those are all protective mechanisms when weaponized correctly. But in the case of sepsis, we're weaponizing it absolutely everywhere, which is just not good. And in the case of autoimmunity, we're weaponizing it against our own cells. And the stimulus, if the stimulus is our body's own cells, well then that stimulus is everywhere because the setting is your body. So very detrimental and very positive feedback loop oriented. So if we think back to PAMPs being read by PRRs, secreting AMPs. So that's pathogen-associated molecular patterns being read by pattern recognition receptors on your innate cells. And then the ultimate, whenever those PRRs bind those PAMPs, that causes release of these AMPs, the antimicrobial peptides. This arc is conserved throughout innate immunity and even conserved in adaptive immunity, just with a couple, extra specific markers there. In the case of autoimmunity, though, let's think about this. So now my body deems some sort of self-antigen to be pathogenic. So I bind the self, and then I release compounds that target the self, which then cause damage to the self. So now I have damage-associated molecular patterns on top of the quote-unquote pathogen-associated molecular patterns when not really. I mean, they are my own antigens, but my immune system's confused at this point. So I have this stimulus getting read, causing secretion of more damaging compounds, causing more of this. Input causes output, causes more input, causing more output. That's a positive feedback mechanism. So I wanted to really hammer this home because most autoimmune conditions are not curable. We have treatments in place. There are symptom management strategies we can employ, but these are chronic conditions that persist because of this inherent positive feedback mechanism at play, because the stimulus is your own body. So, yeah, very, very unfortunate mechanism at play. So let's talk about tolerance. All right, what is tolerance? Well, it's the immune system's ability to ignore the self and commensal bacteria while remaining vigilant against anything pathogenic. Okay, again, tolerating the self. If those cells that are taking care of my physiology should be left alone because they're keeping me alive. Commensals, yes, they're bacteria, but they should be left alone because they're helping me digest. They're helping me with vitamin synthesis. They're helping fortify my gut membrane. whatever the function occurring is, it's not harmful. It's actually helping my physiology, so we tolerate it. The moment it becomes pathogenic, we get vigilant. We say, get out. All right? We want only things helping correct physiology at play. So how do we maintain tolerance? Two things, central and peripheral tolerance. Central is what we talked about in Unit 2 with the B cells and Unit 3 with the T cells. This is established in the thymus for the T cells and the bone marrow for the B cells. This is when immature lymphocytes that recognize self-antigens too strongly are eliminated through negative selection. So that was the process in B cells, the second checkpoint, where we test, hey, is this B cell receptor binding bone marrow? Is it binding blood cells? Is it binding anything that has to do with me? If so, bye. In the thymus, this was, again, checkpoint number 2, negative selection with the medullary thymic epithelial cells. We had the expression of the enzyme AIR, autoimmune regulatory enzyme, yes. If the antigens we present to that budding T cell are from the self, and that T cell reacts to those self-antigens, then it gets the axe, bye-bye. That whole concept was called central tolerance. So establishing self from non-self at T cell and B cell college is central tolerance. Peripheral tolerance is the idea, well, some self-reactive lymphocytes are going to escape. Nothing is perfect, especially in biology. So we rely on peripheral regulators outside of the central primary organs, your bone marrow and your thymus. So those are your T regulatory cells. They ensure that any lymphocytes that were self-reactive that escaped, we calm them down. And I say these ensure tolerance occurs, but sometimes it fails. And in the case of autoimmunity, we get failure on both of these fronts. For some reason, these lymphocytes that were self-reactive did not get eliminated through negative selection. And for some reason, the T regulatory cells that were meant to get rid of the stragglers are slacking as well. So in cases of autoimmunity, we have a breakdown of central and peripheral tolerance. So how does that occur, or why does that occur? A couple things: genetic, environment, or just random. So this does not explain it all. There's a lot of research going on here. We do have some genes associated with autoimmune conditions, and your environment does play a role, but there's more to be discovered, to be sure. But let's look at the genes. I don't have everything. I don't have all the genes for you, but I think this gives you just an idea of where some of these predispositions come from. Certain HLA alleles present self-peptides in a way that inadvertently or accidentally activates autoreactive T cells. How this happens is a bit too biochem for me. All you need to know is that your HLA alleles, that's what coded for your MHC 1 and 2. Your MHC presents peptides to your T cells. So how this HLA is read is how your MHC presents. So if that presenting pathway or that presenting mechanism is a bit different, that can have a variety of effects, right? And we saw that when we talked about HLA back in Unit 3. There are some combinations that are better than others because we see some combinations are not in the gene pool anymore. So that means natural selection selected for combinations that are better. If there are combinations that are better, there are combinations that are a little worse. And some of these HLA alleles have been associated with being a bit worse, being more likely to bind self-peptides and activate autoreactive T cells. B27 is common. Not guaranteed, not 100%, but we see it in type 1 diabetes. And these others, I forget, I apologize, but we will talk about them as they come up. And I'm not going to have you guys remember these by any means. I want you to understand the concept that HLA codes for the peptide presenter, the MHC. So if that HLA allele is a little bit weird, like in these cases, that can lead to autoreactive T cells. Again, not a guarantee. You can have these HLA alleles and produce no autoimmune response your entire life. Not a guarantee. It is an increased likelihood, an increased risk should you have these genes. And again, just a mutation in the tolerance regulators. A big enzyme that we talked about was autoimmune regulatory enzyme, and you should absolutely remember and know it for this unit as well. If that enzyme is not functioning properly, then my T cells that are autoreactive will, that number will increase dramatically. Environmental triggers. So again, some folks might have the propensity, the potential to generate an autoimmune condition, but these stimuli might push them over the edge when they maybe didn't have an autoimmune condition beforehand. So things like infection or toxins. So some bacteria or some viruses we've seen can really dampen the immune response or mutate the immune response, which might lead to autoreactive conditions. Tissue damage can absolutely lead to autoimmunity. We'll actually talk about that in just a moment. And UV exposure, right, that's solar radiation that's going to cause DNA breaks, which can lead to DNA mutations. So when we think of, melanomas, that's certainly the case. And then stress. Stress is the number one killer, right? I don't have a science paper to back that up, but stress is just not good. I'll leave it at that. And that really does decrease your immune response, the efficiency of it, and its ability to differentiate and express correct tolerance. Okay? So, yes, your environment certainly plays a role in the sparking or the initiation of an autoimmune arc. There is a hormonal aspect to it as well, and I'm going to be very brief on here because I'm out of my depth. All I know is that autoimmunity is 3 times as likely in women. So that would suggest that estrogen has a role here, and it has been shown to enhance B-cell survival, but it really only partially explains this discrepancy because men have estrogen as well. So what's going on here? I don't know. We all know women live longer than men. Maybe that is skewing the curve here. I just don't know. But that is a discrepancy that I find interesting that I wanted to make note of. So how does autoimmunity occur? We had a couple examples there, but okay, let's go back to how does infection or how does a bacteria releasing a toxin, how can that cause autoimmunity? Molecular mimicry is a really, really good example of this. So this is when a foreign antigen closely resembles something that you produce. So if an antigen just looks a lot like your pancreas or looks a lot like one of your nerves, who knows? Once lymphocytes are activated against that pathogen, that produces, you could potentially have a cross reaction with the host tissue that share that similar peptide motif, that similar peptide signature. So if we have some similarities between this foreign antigen and the peptides that make up myself, we could potentially have cross reaction. This is a testament to the fact that there are only so many organic compounds that we can produce in nature, and we're bound to have some overlap here. Unfortunately, that overlap can lead to autoimmune conditions. Another case is bystander activation. So during infection, specifically like chronic infection or tissue injury, inflammatory cytokines will activate nearby antigen-presenting cells. And these APCs can accidentally present self-antigens in a co-stimulatory context. So when I say co-stimulatory, I'm referring back to not only do you have CD4 matching with MHC2, but you also had the CD28 matching with B7. This only happened when there were PAMPs present, the B7 pathway. And the reason for that was to ensure that whatever reaction we were doing, whatever we were reacting against, was causing damage to us. Because sometimes we'll bind the self, but if I don't also have pathogen-associated molecular patterns with it, then we know not to cause a reaction. And that was the whole point of the B7 co-stimulation. But in cases of chronic infection and tissue injury, B7 is always up because we're having DAMPs and PAMPs in the area, so that causes this to be expressed. So we could get co-stimulation when it had nothing to do with whatever this antigen was. So if that was me, if that was my own tissue, then I get this bystander activation despite the true inflammation not being related to the self tissue, if that makes sense. So you kind of just get caught in the crossfire. Like that's why I have this guy here. There was no response going against this guy, but since he was just near the collision, he got picked up and a response was produced against him. Him being your own tissue. Then another concept is epitope spread. So tissue damage can release new self-antigens that were not previously available to the immune system. So this is a great example of when immune privilege is breached. The best example of this is a condition known as sympathetic here we go. Ophthalmia. I'm completely butchering that, but ish. This is the case where you have a blowout fracture in the orbit, maybe like a boxing accident or a motor vehicle accident. And now the antigens of the eye, just the proteins of the eye, are floating in the blood. The eye is separate from the body's immune system because we want to separate that. If I had a full immune reaction to some antigen in my eye, well, that would cloud my vision and my eye would be useless. Okay, so the immune system doesn't know my eye exists. With the blowout fracture, now it does. And now I have a full response because those antigens were previously not available to my immune system, so I wasn't able to learn that they are part of the self profile. So now we have a big reaction against the ocular antigens because the body is now aware of something that it deems to be foreign. So we can have that spread. So I have the forest fire here because if you think about it, we're usually fighting bacteria that are fairly small, unicellular little beasts. But if my body thinks my entire eye is a problem, right? That's like, that's 10,000 times bigger than a little unicellular bacteria, right? So the immune system says, oh, that's a huge problem. We need to get rid of that ASAP. So that's another case of autoimmunity, just that spread of an antigen that, a self-antigen that we didn't really know existed from an immune perspective. Okay, and lastly, just to kind of broaden out why this is occurring, from an evolutionary point, right, we evolved for maximal pathogen flexibility and recognition. Okay? So we cast a wide net. Unfortunately, that wide net will inevitably produce occasional cross reactivity with the self. Again, there's only so many peptide motifs in nature. We're bound to overlap. We're bound to mess up. That's built in to immune diversity. That's fine. It's going to happen. Regulation's going to overlap will happen, but regulation should control it. That's the idea. So this mild autoreactivity, it's fine, but that's why I have T cell and B cell college. That's why I have T regulatory cells. So autoreactivity, and it's kind of like cancer. The mistakes in biology happen all the time, but you have corrective and regulatory mechanisms in place to hopefully catch it. When we don't catch it, that's when we have autoimmunity. What's the epidemiology here? So 5 to 10% of the global population and rising. That's a pretty stark number. 78% of these conditions occur in women. Again, crazy discrepancy there. Usually rising in ages 20 to 50. So more prevalent in urban and industrialized areas. Again, hearkening back to that hygiene hypothesis. Maybe we're too sterile and we're knocking out some potential commensal bacteria that would cause my immune system to calm down and not be autoreactive. Maybe I wasn't exposed to enough germs at a young age to properly gauge my immune system so it knew what self was and what actual threats were. Who knows? Again, I'm not trying to have you guys be unhygienic, but there's a lot of good thoughts behind the hygiene hypothesis, and I don't know what the right answer is. But still, shower and wash your hands, please. Prevalent in regions of higher latitude with less UV exposure. We'll certainly see that with MS. So yes, UV light and UV radiation is correlated, with increased rates of melanoma. Right? If we're not wearing sunscreen, increased DNA breaks, you're running the risk of developing melanomas, carcinomas on the skin. That's not to say sunlight is bad for you, though. Right? Sunlight in general is very helpful because it initiates that vitamin D synthesis pathway. And you have a lot of calcium-dependent biochemistry going on at the immune level. So sunlight in and of itself is very, very important for correct immune modulation. Yes, UV radiation can be bad. Just don't go outside if the UV index is high if you're super worried about that. Sunlight exposure is very crucial for proper immune health. Now, is that to say if you're not getting sunlight, you are going to get a autoimmune condition? No, absolutely not. But low UV, low vitamin D is absolutely correlated with these diseases. Doesn't cause it, but it is a relationship that we have noted. So it's something to consider, is all I'm saying. MS, celiac, and also Crohn's. So that's just IBS. Many flavors of IBS. These are all autoimmune diseases, and they are on the rise. And it's really interesting as to why. A lot of good theories out there, and I'm sure there's going to be a lot of great research that comes out in the coming decades around them. But yes, that is the primer for autoimmunity, and we will talk specifically about gastrointestinal autoimmunity on the next talk, and neuronal autoimmunity, the talk after that. So thank you for your attention. I really am excited about this unit, and I hope you have a wonderful day.